Hello and welcome to this screencast. We will be addressing the microanatomy of the retina. And so again, here we're looking at the microanatomy of the retina and then a little bit about the photoreceptive cells, the rods, and the cones. And we'll try to talk about the rhodopsin, the role that rhodopsin plays in vision. And I think we'll do a little bit more of this in class later on. So first, microanatomy of the retina. So here we are at the posterior part of the eye where the optic nerve is leaving the eye. So if we zoom in on that square right there, that gets us to over here. And we see the optic nerve leaving the back of the eye. These are sensory fibers of cranial nerve number 2, the optic nerve. Right? And so these are going to go back to the thalamus, and then from the thalamus, tracks lead back to the occipital cortex where vision is processed. So we see that there is this layering going on of the sensory tunic of the retina itself. We also see that there are blood vessels that are going to be associated with this optic nerve as well. So let's zoom in here on the neural layer of the retina, and we see that there are then layers of cells. So this is the more posterior aspect of this, and this is more anterior. So the vitreous humor would be right here. The pathway of light is coming from this direction. If we go back, here's the choroid layer, and then there's this pigmented layer of the retina, and this pigmented layer appears black and is to prevent any scattering of light waves that are going to be coming from this direction here. So what's happening is the photoreceptive cells, the photoreceptors, are the rods and the cones. So these are photoreceptors, and we'll talk briefly a little bit about those on the next slide. But these photoreceptors, these rods and cones, are modified neurons, and then they will synapse with bipolar cells. So these bipolar cells are not unipolar, they're not multipolar, they are bipolar. So the cell body is right here. So there's this synapse. So the rod or the cone is going to synapse with a bipolar cell. That bipolar cell will then synapse right here with a ganglion cell. So the ganglion cells are this third layer. So we have roughly 3 layers of cells: the photoreceptive cells, the rods, and the cones, this layer of bipolar cells, and then the third layer here of the ganglion cells. Now, interspersed in this middle layer of the bipolar cells, you have these other cells here that are connecting and communicating. Right? So this cone here would be communicating with this rod, communicating with this rod over here, etc., and then also communicating with this bipolar cell. So this gets complicated, complex in a hurry. But those 3 layers are visible under the light microscope, and I encourage you to go back and get a slide and look at them under the microscope. So a little bit more about these ganglion cells then. These ganglion cells, here's their cell body, here are their dendrites, and then the axon of this ganglion cell here comes here and it makes a 90-degree turn and then is going to fuse or join with the other axons of the other ganglion cells. Right? And so these axons, these whole collections of axons, are going to continue, and then that's what you're going to see right here. They continue, and then these are all axons, and then these axons leave the back of the eye and become the optic nerve. And this makes sense to you because you know that nerves are collections of axons in the peripheral nervous system. And since this is the optic nerve, this is making sense to you. Now, what's crazy, in my opinion anyway, is that this light comes in to the eye, light waves come back here and strike pigments that are in these rod cones, and they strike pigments that are in these rod cells and in these cone cells. And then the pigments that are in these cells will in some way generate action potentials that are then transferred via the synapse to these bipolar cells, transferred that action potential via this synapse to these ganglion cells, and then the action potential on these ganglion cells is conducted all the way down this optic nerve. Right? Under the light microscope, here we go under a scanning view, and so you'll be able to see this very clearly on the slides that we have back there. So I encourage you to take a look at this next to what you're looking at under the microscope. So you're scanning, Here it is labeled for you so that you can then sort of see what's what under the light microscope there. This is a little bit more detail than what you need to know in terms of like all these outer layers and inner layers, you know, ONLs and OPLs, etc. But just look for basically, right, so here are the photoreceptors, the rods and the cones, right? This is the bipolar layer, and then you have the ganglion cell layer, right? And so when you look into the light microscope back there, it'll look very similar to that. So again, here we go, right? See if you can identify ganglion cells, see if you can identify this internuclear layer, right? Here's the OPL, ONL. Again, I'm not having you, know this much detail, but again, looking at these and being able to identify them, you might, if you have an oil immersion scope, take the oil and try and look and see what it looks like here. Let's see if you can get these rods and cones to resolve a little bit better there. They're really jammed in there, so take a look at that. Now, in terms of the rod and the cone itself, let's look at the rod cell here. So here's our rod. This is all one cell, and it's a modified neuron. It's modified in that this end of the cell right here has these folds. So if we go and look right here, this is the membrane of the cell, and we see that that membrane is highly folded, right? So highly folded membranes increase our surface area. And if we are increasing the surface area, we can jam more stuff in those membranes. So what's in the membranes, if we now look at what's in the membrane, so here's our phospholipid bilayer, right? And we see that we have this pigment called rhodopsin. Rhodopsin is the name of the pigment, and it has two subparts to it, opsin and retinal. Retinal is derived from vitamin A, which you find in carrots. So if you hear people eating carrots and it improves your vision, you're getting vitamin A, so you're able to make more retinal. This is kind of the logic behind that, but I'm not so sure that that is exactly, true. But I'm sure that I'm sure there's some truth to that, but it's not, straight one-to-one correspondence there. So, but anyway, back to this rhodopsin. So here's rhodopsin, and it has this opsin and retinal. Now this rhodopsin is a pigment, and because it is a pigment, it is able to absorb photons. It's able to absorb wavelengths of light. So those of you that are in AP biology or had AP biology, and we talked about photosynthesis and absorbing photons, chlorophyll is something that's very similar to this. There are a few tutorial videos that I found on YouTube that I posted on Blackboard. If you want to go and take a look at the super biochemical details of this, by all means, please do so. It's actually pretty interesting. But essentially what's happening is that when this pigment rhodopsin absorbs photons, this opsin and retinal change their shape. Right? They change shape, and that shape change here, don't worry about all these G proteins and things, this shape change right here is linked to then a sodium channel actually closing. Right? In the cones, right, the sodium channels will open. But so again, let's go back and look at this again. So as the pigment rhodopsin absorbs light, it changes the shape, and that shape change then will close these sodium channels. So if you close sodium channels, you actually send no action potentials. You don't depolarize this cell to threshold. Well, wait a minute. So that means that this cell actually was already depolarizing. So its normal state when there is no light hitting them is that they're actually depolarizing to threshold, and they are generating action potentials. So they're releasing neurotransmitters here. So now when light shines on them, these rods here, you get this absorption of the light, you get the shape change, and you get the sodium channel closing. And the sodium channel closing then stops any transmission at this neuron right here, and that actually then depolarizes the bipolar cell, which depolarizes the ganglion cell, and then you have that information in some way transferred to the, or interpreted at the occipital cortex as light. If that sounds confusing, one, it is a little confusing. Two, we'll take a look at a little bit more detail in class, and I can go over that a little bit more specifically with you. But this is essentially the rods and the cones, right, what's going on there. So just to go back and again take a look here, we talked briefly about the role of rhodopsin, rods and cones in these photoreceptive cells, and then the microanatomy of the retina. Right?